Welcome to The Explainer. Today we're going to tackle a really important topic, testicular germ cell tumors. We're going to walk through the whole journey, right from that initial discovery of a mass all the way to the major decisions that shape a person's treatment.
All right, so let's put ourselves at square one. A testicular mass has been found. The first question on anyone's mind is going to be, okay, what happens now? And that one question really, it kicks off a very specific, well-defined diagnostic process. Okay, so let's get into it. The process starts with three really critical steps. First up, they'll do an ultrasound, a USG, to get a good, clear picture of what's going on. Then, it's on to blood tests. They're looking for what are called tumor markers, specific proteins that can signal cancer. And the final step? It's a surgery called a radical inguinal orchiectomy. Now that sounds complex, but it basically means the entire testicle is removed through a small incision in the groin. And this is key. It's not just a diagnosis, it's actually the first step of the treatment itself. But it's just as important as what they do, it's what they don't do. With a lot of other cancers, you'd expect a biopsy, right? But not here. A biopsy straight through the scrotum is a big no-no. And the reason is so important. Doing that could literally open up new highways for cancer cells to escape and spread. It's a huge risk. That's why they go through the groin. It keeps everything contained.
So, once the orchiectomy is done, that tissue goes straight to the lab. And what the pathologist finds under the microscope, well, it leads to a massive fork in the road. Because it turns out, pretty much all of these tumors fit into one of two major buckets. And which bucket it's in, that changes everything about what happens next.
And here it is, the core of our whole explainer. On one path, you've got seminomas. These are the most common type, and the cells look very uniform, almost like clones of each other. Then, on the other path, you have the non-seminomas, or NSGCTs. This is more of a mixed bag, with a bunch of different cell types all jumbled together. The differences between them, how they look, the markers they produce, and most importantly, how they respond to treatment, this is what creates those two totally separate journeys. All right, let's head down that first path. We're going to look at the classic seminoma. This is, you could say, the more common and straightforward of the two.
So if you were a pathologist looking through a microscope, these classic seminoma cells would really stand out. You'd see these sheets of cells that all look pretty much the same. They're big and round, they have this clear, watery-looking stuff inside them, and a big nucleus right in the middle. It's all very neat, very consistent.
Okay, here's a quick but important little detail. We have these rules about tumor markers, right? But biology doesn't always play by the rules. While we usually think of seminomas as HCG negative, in about 7 to 15% of cases, they actually do produce some HCG. It's a known exception and something pathologists are definitely looking out for. Okay, let's switch gears and go down that second path, the non-seminoma group. And this one's totally different from the seminoma. It's not one single thing, it's actually a whole collection of different kinds of tumors. And sometimes you can find a mix of them all in the same place.
And in this group, you've got a few major players. There's embryonal carcinoma, there's the yolk sac tumor, which is the one we see most often in babies. Then you have choriocarcinoma, which is known for being super aggressive. And then there's the teratoma, which is kind of wild. It can actually grow different types of tissue, like hair or even cartilage. Just fascinating stuff.
And speaking of that yolk sac tumor, it has this really cool microscopic signature. It's called a Schiller-Duval body. It looks kind of like a tiny, primitive organ, a blood vessel surrounded by tumor cells. When a pathologist spots one of these, it's a dead giveaway. It's like finding a fingerprint at a crime scene. They know they're dealing with a yolk sac tumor. This table really brings it all together. Remember those blood tests from the beginning? Well, this is why they're so powerful. It's basically a cheat sheet. The two markers, AFP and HCG, create these unique patterns. For example, if AFP is high, you're probably looking at a yolk sac tumor. If HCG is through the roof, you're thinking choriocarcinoma. It's a huge help for confirming the diagnosis and even for tracking how well treatment is working later on.
So once the team knows what they're dealing with, seminoma or non-seminoma, we move into the final phase. Now it's all about staging. That just means figuring out if the cancer has spread anywhere else in the body. And once they know the type and the stage, they can build the right treatment plan. In the general flow, makes a ton of sense. Step 1, the orchiectomy, which gets rid of the main tumor. Step 2, staging scans, usually CTs of the chest and abdomen, to hunt for any signs of spread. Then, step 3, you put all that info together, the tumor type, the stage, and you get a custom-built treatment plan.
And this is where that fork in the road we talked about becomes absolutely critical.
For early-stage cancer, the two paths couldn't be more different. Seminomas, they are incredibly sensitive to radiation. It just works really, really well. But non-seminomas? Not so much. Radiation doesn't have the same effect. So for them, the options are a really big surgery to clear out lymph nodes. That's the RPLND, or chemotherapy.